broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and welcome to this webinar today. My name is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. Today's webinar is presented by ASCCT and also by the European Society for Toxicology In Vitro. And if you're not familiar with either of our organizations, I encourage you to go to our websites to learn more about us. We have a lot of activities, annual meetings or biannual meetings, but also activities like this throughout the year, training programs, and support for toxicologists in these fields, and early career scientists and students as well. So just some general housekeeping first. The webinar is recorded, and the recording will be posted on the ASCCT website in a few days. Also, the lines are muted. There will be an opportunity to ask questions after each presentation. You can see in the GoToWebinar control panel a little question module or menu. Type your question into that question box, and I'll read them out during the Q&A. And again, we have two presenters today, and we're going to have two Q&A sessions, one after each presentation. So go ahead and put your questions in while the presenters are speaking, and we'll be able to ask them. I also want to mention a couple of upcoming meetings. Upcoming webinars. In January, we have award winners from our ASCCT meeting, which was in the fall. And you can see on the top of your screen, you can go to our annual meeting webpage to find out what happened there. And these student award winners are going to be presenting their work on January 14th. And you should be able to register on the website for that webinar very soon. We're actually switching platforms with the new year, and so we'll get that up there soon. Also in February, we have almost scheduled very exciting PBK and QIV-IVE, quantitative in vitro to in vivo extrapolation webinar, and so you'll be able to register for that as well. If you would like to sign up for our email list in order to find out when webinars are announced, please do that. You can do that on the website. I wanted to mention our partner society, ESTIV, is having its next meeting in 2022, but there's lots of things going on in that society until then, so I would encourage you to visit the website to find out more about their activities. And also, QSAR workshop, the International Workshop on QSAR and Environmental and Health Sciences, was postponed to June of 2021, and that's going to be virtual. And you can now submit your abstracts. Abstract submission is open until the 30th of January. So today we have two presentations. I'm very excited for both of these presentations, and I'm very grateful to Dr. Wolf and Dr. Coleman for coming to speak with us today, because we were able to schedule it a little bit really quickly on short notice, and they stepped up and prepared these presentations, and hopefully you're looking forward to it as much as I am. So without further ado, I'm just going to introduce our first presenter, who is Michael Wolf. Dr. Wolf is a distinguished scientist and back-end fellow at Medtronic, where he's worked for 32 years with a focus on medical device biocompatibility and blood material interactions. During his tenure, he contributed to the development and assessment of new technologies and helped in the evaluation and release of numerous new products. Wolf is also convener of the ISO 10993-4 international standard that focuses on medical device interaction with blood. In this capacity, he guided the development of the 2017 revision to that standard. He has a number of patents, peer-reviewed publications, and book chapters, and serves on the International Society for Applied Cardiovascular Biology Executive Board. Mike has BS degrees in biochemistry and in chemical engineering from the University of Wisconsin-Madison, and a master's degree in biomedical engineering from the University of Minnesota. Michael, the floor is yours.
Okay, thank you, Christy. I'm going to show my screen. Can you see anything at this point?
Yes, we can see your slide deck.
Okay.
Great.
Super. Okay, minimized again. Okay, good morning again, Christy and Kelly, and good morning and good afternoon and good evening to all of you who've signed into this webinar. Thank you for joining us, and very happy to share with you some of the work that I'm doing in what we call the Medtronic Operational Headquarter Blood Lab, located in Minneapolis. And what I'm going to do over the next 20 minutes or so is just to share with you some of the work that we've been doing to try to expand and better develop some of the in vitro methods for assessing the thrombogenicity of medical devices and medical device materials. And I think you'll come to appreciate through the talk that we're trying to apply what we call a use condition and approach to this, because there's many issues that have gone on in the area of blood testing over the years, and there's many cases where people have failed to really try to mimic the use conditions, and so that's really what has been the focus of the work in our lab. I suspect that there are at least a few toxicologists out there, and I suspect you recognize this fellow here. I like to start off with showing Paracelsus, and one as a reminder to mention again that I'm not a trained toxicologist. My background is actually in biochemistry and chemical engineering and biomedical engineering, but this phrase that Paracelsus is credited of saying the dose makes the poison I think is maybe challenging to some people who are in cardiovascular medicine or in terms of toxicology. What does it mean, the dose of a medical device? And I think if Paracelsus was with us today and had been studying thrombosis, I think he would agree that the blood contacting surface area, the surface chemistry, the device geometry, fluid dynamics, the type and amount of anticoagulation, the arterial venous vascular position, and other things such as patient genomics and comorbidities, these all make the poison, and the poison in this area of thrombogenicity is thrombus itself, which can interfere with the device performance, and also equally bad is if it leads to some complication with the patient. So for anybody studying this or working in this area, this is one of the main documents on their desktop, this ISO 10993 Part 4, selection of tests for interactions with blood. This is a horizontal standard, meaning it has to cover all blood contacting devices, so it's been a very difficult document to pull together. But it's a document that applies to all new cardiovascular devices, new device materials, and any change that happens to an existing device. And it really only applies to applications that involve direct or indirect blood contact. If there's no direct or indirect blood contact, you don't need to have this document on your desktop. And this just gives you a hint of the kind of devices we are working with at Medtronic. This is a very small fraction of the blood contacting devices throughout the company, and actually we have estimated that over half of our products do have a significant blood contacting surface. But what you see, and I'm not sure if you can see my pointer here, but up in the upper left is a very tiny coronary stent that maybe is a half a square centimeter. Other tiny devices, and that's about the actual size of this micro pacemaker that gets implanted in the right ventricle. That very large looking device in the upper right is a blood oxygenator that just the white fibers within that shell are 25,000 square centimeters. Here, this heart valve that's shown in the sort of in the middle of the screen is made out of ceramic, pyrolytic carbon, and a fabric. The TAVR valve next to that is metal and biological tissue. This device that's shown spanning the neck of an aneurysm actually goes on the arterial side of the brain, which is a high risk area for a medical device to be. And these other products are other examples. Here's an ablation catheter. This weird apparatus down here on the bottom is a console for renal dialysis, and the other one next to it is for pediatric applications. And then way in the lower right corner, you see one of our newer devices that's in the Medtronic portfolio, a heart ventricular assist device that's a permanent implant device. And the kind of things we're trying to avoid and the standard is trying to teach is thrombosis associated with these devices, where it happens even in long-term heart valve implants. Actual tissue can grow up over the device and cause weird flow dynamics that can lead to emboli shedding. There's a lot of catheter technology today, and you see catheters in the upper right and in the middle of the screen. These devices perform pretty well even when thrombus forms on them, but if thrombus occludes the delivery lumen, then they will not function properly. Same can be said for cannula, which is shown in the lower right. Their occlusive thrombus will actually make that device fail in the field. And then in the lower left here, you see two sensors implanted in the right ventricle. One sensor has failed. It's become completely encapsulated with tissue, and the other one is working fine, but you can see sort of on the base of that device is a brown material, and that's organizing thrombus that is eventually going to probably grow up over that sensor. So the main place that people open to in this particular standard is this table 1, which shows the test categories. It's divided by hemolysis and thrombosis areas, and thrombosis itself can be divided into in vitro and in vivo tests. There's quite a bit of opportunity for in vitro testing in the external communicating devices, but as you look down into the more permanent implant devices, you see that it looks like there's only in vivo or ex vivo applications that apply there. But it's important to point out that people work very hard at putting in this little bit of footnotes here, and I'm going to expand that so you can see it a little better. But importantly, it points out that thrombosis is an in vivo or ex vivo phenomena, but it can be simulated with in vitro conditions. So in vitro, in vivo or ex vivo testing might not be necessary if clinical relevant in vitro thrombosis testing is performed. So for those of you out there who are involved in this, it's important to point out that there is an in vitro opportunity here, and I'll give some examples of that at the end of this talk if there's time.
Mike, I'm sorry to interrupt, but just for a moment, your audio keeps kind of getting quieter than louder, and I wonder if you're turning away from the microphone or something like that.
Okay, let me pull the screen a little bit closer and I'll continue and tell me if it's sounding any better.
Okay, thanks.
So the standard leaves it up to the manufacturer to determine which of these important in vitro tests are appropriate and if they're a reasonable alternative to in vivo testing. The next most important table in the document is table 2, which lists the common tests used to assess interactions with blood. For those of you who are familiar with earlier versions of the standard, you appreciate that this table is now much more simplified and much easier for people to digest and use. However, I just want to take this one moment to point out that these standards are not set in stone and they're actually always evolving. And while we came out with this revision in 2017, we already know of areas for improvement. For example, in thrombosis, gross analysis testing is still heavily used, and one particular model, the non-anticoagulated venous implant model, is fairly controversial still and problematic. And I would encourage people to read and take a look at those two references there, which show a lot of the pros and cons of using this model. The other problem with these common tests is there's not very many that are well documented. For example, this PTT assay is fairly well documented, and I'll cover that in a little bit. And this other simple in vitro test, platelet count test, is also fairly well documented in this other particular standard referenced here. But the PTT test is a very simple test, and it only measures time for clot to form. And it's a very simple test. It takes 4 to 6 square centimeters of material and uses citrated blood, which is not without its problems. And the test it usually involves a positive control, negative control, and a legally marketed comparator device control, and a blank control. And everything's done in triplicate. And after a certain amount of exposure and the addition of this thing called rabbit brain cephalin, and then the addition of calcium are two big steps that make the assay work, but it's a very simple test, and in the end you end up with the amount of time it took for a clot to form. And the other test is also very simple. It only measures platelet counts. It's sort of a misnomer. It's called a platelet leukocyte count test, but in the standard, it admits somewhere towards the end that the leukocyte data workup is not always necessary, because there hasn't been any validation done to the interpretation of that part of the test. So people really only use it to look at platelet count. And this test is also incredibly simple, but here it actually calls for 12 square centimeters of test material per mil of blood, and it calls for, again, using citrated human blood. And in the recent edition, they've added the opportunity to use heparin instead of citrated blood, which is a huge improvement. But again, the layout is similar with the various controls. And in the end, the counts are done with a standard Coulter counter. The problem with this test, and it's kind of illustrated in this schematic, is for a simple device like a stent there that adds about a half a square centimeter to a square centimeter, that gets exposed to the whole body volume of blood in an adult, which is about 5,000 milliliters. So when you work out that exposure ratio, it's 0.002 square centimeters per mil. You take the next device, which is also very small, the micro device, that's 5 square centimeters, and still a very tiny exposure ratio. Then the next device in the middle, 50 square centimeters, is a typical surface area for a pacing lead. That leads to 0.01 square centimeters per mil. But then when you get into these bigger devices like a renal dialysis cartridge or a blood oxygenator, these are very large surface area devices that have upwards of 15 to 25,000 square centimeters, and that leads to about 5 square centimeters per mil. But then you come back to this ASTM standard, and they're asking users to test 12 square centimeters per mil, and that's roughly a 60,000 square centimeter device, which no such thing exists on the planet. So the test is prone to false negatives because of this grotesquely large exposure that's being called for. So what we've done in my lab is to look at some of these other tests that are in this in vitro category, and we specifically look at thrombin generation, fibrin formation, more sophisticated platelet count testing, and platelet activation using this beta thromboglobulin protein. And where do these things come from? Well, they come from the coagulation cascade, which you can see here way in the upper left. When platelets bind to a surface, they release this protein beta thromboglobulin. So that's a good measure of platelet activation. When thrombin forms, this small fragment F1.2 is given off, and there's a bioanalytical assay for that fragment. When thrombin causes fibrin to form, there's a small fragment given off called fibrinopeptide A. We can measure that. And also coming back to thrombin, when thrombin is activated, there's an endogenous protein called antithrombin that binds to that, and there's a bioanalytical tool to measure that as well. And this is just some pictures of those kits, these ELISA assays that are commercially available. They're approved for clinical use. They're used routinely in hematology labs, and they've just been adapted in our lab to use in medical device testing. So I'd urge you to, if you have time, to go get a copy of this paper. We've paid the fee that allows everybody to take a look at it for free. And this is a summary of the work that we've been doing in our lab to test these various tools. We have our own standard blood collection tube, test tube model that we report on in this paper, and we compare it to what we call a circular test tube or a modified closed loop test tube, where blood actually rotates around in there in a pulsatile fashion. And this slide just shows you all the things that go together to make the test tube model. There's a lot of things that have to be pulled together. But in the end, what we do is we take a non-additive Becton Dickinson blood tube, remove the vacuum, put a small quantity of anticoagulant in there, put test materials in there, recap it, and recreate a vacuum. And then we just use standard blood draw techniques to fill the test tube. And I'll just show you how that works. So that needle is hooked up to the donor arm, and it's just like what we see in the clinic, how we all donate blood samples every day using this vacuum technology. And it just took a while to work out this precise method to pull the exact volume that pulls in the exact volume of blood. So that's the blood filling by vacuum draw. We have another method that's called saline displacement. And I'm muting this so you guys don't have to hear us chatting in the lab. But this is a simple method where a syringe is hooked onto an epidural needle that sucks out a certain volume of the tube. And as it's sucking out volume, it's pulling in the same volume of blood. And so the test samples in the tube immediately get exposed to blood, and there's no air interface in this model. So test materials are immediately exposed to fresh human blood in these various models. Here's the layout of the test loop model, showing all the various paraphernalia that goes on into doing this kind of testing. Here is an example of how we fill this test loop. I'm going to turn off the chit chat in the lab. And I don't know who's all in the audience out there, but that's Bill Teelocker, my colleague, who has very nice veins and always willing to sit down in the chair and donate blood for our causes. And just to give you a feel for the type of rotational flow motion that this particular model calls for, it's this circular movement that involves every time it goes through this pulse, a little check valve closes, and when it pauses, the blood flows forward. So it's very much like a pulsatile flow model. So coming back to this paper then, that starts off with explaining three very large studies that were done that we call material thrombogenicity exploratory studies that took all the same materials in the various studies, but we look at model as a variable, donor as a variable, exposure time as a variable, and heparin level as a variable, and then we follow up with these various bioanalytical tools. And I know you guys call this group computational toxicology. Well, these three studies, for your information, were full factorial studies, and this shows the experimental design. So if you look at these, each study is a substantial amount of work, and I can tell you that under the materials, everything was in duplicate. So just imagine in material in this MTES1, there are 12 tubes times 3 donors, that's 36 samples times 3 different time points, so that's 108 samples, and then times 2 different heparin levels. So that study in itself took 216 samples to do, and then the next one down, if you do the math, it's 96 tubes or loops to do those as well. So a fair amount of work. I show the pictures of some of the test materials down below. And not too surprisingly, when we do the ANOVA analysis, we find that whether we're in the material thrombogenicity exploratory study 1, 2, or 3, you look at TAT F1.2 and beta thromboglobulin, you see a lot of very highly significant figures. And so not too surprisingly, materials incredibly important. Heparin is very important. Donor and time and model are all extremely important. And I can't tell you how many times that people report on blood studies, and then we see that they used a heparin level of 3 or 10 units per mil. And well, when the heparin is that high, nothing happens. Quite often people do blood studies and they don't look at more than one donor. So they lose out on what happens when you have multiple donors. So these are all important factors that we've come to recognize and report on here. And then we come up with this simple standard design of experiment to test materials using this format. And just let me walk you through this. So here we have glass, polyethylene test material, legally marketed comparator device. You can put other materials in it and always need to include no materials. So we set up all those in the tube or the loop assay and then call donor X to come into the lab, and they do their first blood donation 1 under low heparin. They come back either the same day or the next day and do a second round study with the same test materials at a higher heparin level. And then we ask the donor to come back on 2 additional days just to repeat those studies to get a better N number. And then we repeat that whole thing on another donor to get the kind of statistics we think would show the effectiveness of these various materials. So the other half of the paper really goes on to look at 6 cases that were done at Medtronic, looking at various vascular catheters or polymer-based vascular devices, and even a neurovascular flow diverter using this experimental design. This is the way we've come up with assessing the results. And I'm just going to walk you through just one of these. Let's take a look at the top, this TAT result. We've drawn red lines, which are the cutoff point, where above which we think the numbers are of concern, below which we're not concerned. But if you just look at that score of 3, here's a TAT value of the test material that's statistically significantly greater than a legally marketed comparator device, but less than glass. And in that category of 4, the test device is greater than or equal to the response of glass. So this is a very simple way of assessing whether things are high or low. And the same scheme was used for all the other factors, and those numbers greater than or equal to come from doing Tukey-Kramer on a significant comparisons of all means. So it's left up to statistics to make the judgment. And here is one set of results on one material. This happened to be, the test material happened to be a heparinized material. And just to walk you through this, there's TAT response on the left, F1.2 on the right, and then beta thromboglobulin in the middle. But basically what you see is data distributed from low to high heparin and tube or loop model. And then there were 2 donors, black dots and white dots. And the fundamental finding here is that the heparin material is indeed more blood compatible than its analog that does not have the heparin coating. And you see how it lines up between the no material and the glass and polyethylene. And I think you could probably sense that it looks like the tube model might be a little less sensitive than the loop model, and that basically TAT and F1.2 gives the same results. And the beta thromboglobulin is a little bit more noisy, but it also shows a trend towards the heparin material being improved over the non-heparinized. And here's one more example, and this just happened to be a case where the test material actually failed. You can see test 6 material is higher than the legally marketed comparator device 6A or 6B, and it's a little bit lower than glass, but that was the trend with 2 different donors and at a single heparin level. And the final part of the paper compares all the test materials that were used in those in vitro tests to actual in vivo NABI results. And this table shows you summary of the results that were scored using the standard NABI score scheme. And these are the scores, which green color is passing, red is not passing. And then we compare all of these results to the in vitro scores in this table. So here again, you see the in vivo scores on the left. Here's all the low heparin in vitro scores. And then here's the concurrence in the basically about 7 out of 8 or 88% of the results showed good concurrence. And So we're pleased with these results, and we're not comparing the high heparin because high heparin does not make sense to compare to a NABI test that has no heparin. So in summary, factors important in thrombosis were originally described by Virchow in the 18th century. He coined this term Virchow's triad, or he didn't do it, somebody assigned it to him. But basically, thrombosis is affected by blood flow, vascular injury, and blood hypercoagulability. But there are many other factors that are involved, and there's at least 3 other very important ones. Obviously, I pointed out that medical device materials are important. Anticoagulants, antiplatelet drugs, and antithrombotic therapies all play a big role in results. And then there's a whole slew of device-specific considerations, such as contact duration, surface area, venous versus arterial position, brain versus cardiac versus peripheral placed devices, etc. So use conditions do indeed play a critical role in thrombosis. So Christy, that's it for now. I do have 2 case examples I can go over, but maybe we don't have time. I'm not sure how we're doing.
I think we have some time if you want to take a few minutes to go through them.
I'll just do it really quickly because that's the results I pointed out up to this point were really done in the last couple of years, but here's some work that we're doing recently where we asked the question, can we apply this to other really complicated devices like this cardiotomy venous reservoir, which is a really important part of cardiopulmonary bypass. But unfortunately, it's the kind of thing that many people think need to be studied with an in vivo model. But we asked the question, well, maybe we can we test the cardiotomy reservoir in a simpler method? And so one of the things that some people at Medtronic wanted to look at was, are there other coatings that affect things differently or better than this balanced biosurface coating that we have? And that's applied to the inside of this device. And so we took this device and we looked at that thing that's circled in blue up there on the left. That's called the down tube, and we're able to remove that from the device after it's been coated. And then we're able to cut out a certain section of that. And actually, you see on the right here, we were able to take that cutout piece and actually use Becton Dickinson caps to turn that into a vacuum tube, just like the ones we use in the tube model. And we know exactly what surface area is inside that little segment, so we were able to draw in the specific amount of blood to give the desired 6 square centimeters per mil exposure. And then we were able to use this very simple DOE design of experiment, testing 4 different coatings against the legally marketed comparator device in glass. And I'm not going to go into the results we got, but it very effectively showed that some of the coatings were very effective, and we were able to do this. You can do the math there, 14 times 2 times 2, 56 samples, about 2 mils a piece, so 112 mils of blood. We could never have done this study with animals. It would have been just grotesquely abusive of animals, and so we were able to do that, avoid all that animal work. Here's another device that's very complicated. It's the ventricular assist device that gets sewn onto the heart. Many people would say we don't have an in vitro model that can simulate that. And it would take 1 device per animal, and there would take dozens of animals to test out all the different variations we're looking at to improve this device. But we came up with this in vitro model, and you can't really see it in here unless you look carefully behind each one of those numbers. But behind each number is about 150 milliliter closed loop that has a ventricular assist device. There's sensors, there's temperature gauges, and it's all hooked up to this computer system, so we can actually test with fresh human blood 8 pumps at once. And this is incredibly remarkable because we can go through many variations of things that we're looking at and rapidly get to improvements in pump performance without having to go through herds of sheep or whatever in Minnesota. I think that's it, Christy.
Great, some good additional work and some good additional points. We've got a few questions for you, Mike. So again, if you have a question, just type it into the Q&A box. A compliment. Thanks for the beautiful presentation. And Monica is wondering, can you discuss the in vitro methods available to or under development to tackle more complex phenomena like fluid dynamics or device geometry? And maybe touch on the main limitations in terms of development.
It's a good question. It's complicated, but we rely on many experts across the company to help with things like fluid dynamics. So there's a whole slew of people who are working on complex devices like the ventricular assist device to simulate a heart model that would show the kind of issues that go on in the left ventricle when that device is being used. And it's very complicated. There can be a suction that occurs, and that can complicate things tremendously. And that's where many in vitro models find it hard to simulate, but we're working on that. So, but that involves computational fluid dynamics and experts in that area to help us out. I'm not sure if I answered every part of that question, but that's one answer.
Yeah. There's a few questions about FDA submission or acceptance. Just wondering if you can say a few words about the loop model and whether FDA might be accepting it or what the pathway
Sure.
might be.
Well, there is talk in Medtronic of submitting this paper and this methodology to the, what is it, Kelly, MDDT?
Yeah.
Is it called?
Yep.
So we're inclined to do that, but we've already had several submissions that have been reviewed by the FDA that have primarily looked at the tube model. And we have various reports coming out right now that have used the loop model to look at renal dialysis catheters. I'm not sure where they are in the submission stage and how far they're going to go. But we are already reaching that point where we are submitting this data in summary in vitro studies that have been done in very GLP-like fashion. Not everything that we've done in our lab is GLP approved, but it's all GLP ready. So we're very confident that the methodologies are sound and everything is extremely well documented. And there's even some parts of this we've reviewed with the FDA. The fact that, for example, when we do platelet counts in our test tube model, we do find that the polyethylene control actually comes out differently than in that ASTM platelet leukocyte test. And the big difference between that test and this test is we are drawing blood right out of donors' arms right into exposure to these test materials. There isn't any way to get more fresh and immediate and realistic exposure than that, but we do see that the platelet counts on polyethylene can be lower than what is generally reported in the ASTM standard, which again, it typically uses citrated blood, and we do not do that. It's not something that we consider normal. So I think they are coming around to understanding that these use conditions we're applying are very appropriate, and the test results are very sound.
Okay. And just to maybe, we have a couple of questions coming in about what is currently required. So just to confirm, the NAVI model is the current requirement by FDA, is that right? And some of the other agencies? They
rely on it heavily, and we're all go back and forth on the meaningfulness of that test. But and I didn't show it, but and it's in the paper. But there's another set of gross pictures in the paper that show that the materials in the test tubes have thrombus building up on them, just like in the model. So we call it a test tube NAVI, but the whole issue with the NAVI model, and I think the FDA is coming around to understanding it, is that even highly FDA approved and very fantastic devices fail that test. I don't know if you recall the one picture of the catheter two catheters next to each other. One's covered with thrombus and the other one has no thrombus. Well, the one with no thrombus is heparin coated B Brown catheter that we coated in our lab. The other one is the clinical device that's used in hundreds of thousands of patients annually that has a thrombus on it.
Mm-hmm.
Another picture, and it's in that paper that describes the pros and cons of the Navi model, but there's I don't know if you recall the pictures of the sensors that we were studying that were in the right ventricle. We studied those sensors in the Navi model to see if thrombus interfered with the sensor reading. And we specifically use in our own lab the Navi model because it's so fantastically gross thrombus on things, and all the materials used on that sensor are FDA commercially available, commonly used polyurethanes. So I think people are coming around to understanding it's not the greatest tool, and thrombosis is a normal phenomenon. All hydrophobic polymers, thrombosis can be mitigated by surface modifications, and so the model is fairly problematic.
Yeah. Well, I will say you're getting a lot of compliments on your work in the chat and the Q&A, and I'll send those to you. I would just echo all of that. I think it's this is how these models are replaced, right? Is by sharing your work with the scientific community and then having others take it up and support it. And so thanks for doing that.
My pleasure.
So I think we're going to move on to Dr. Coleman. So I will introduce him and make him presenter. Or yeah, Bethany, if you want to go do that, that's great. Thanks. So Kelly Coleman is a distinguished toxicologist at Medtronic, where he provides preclinical product development support. He is a U.S. expert on the ISO 10993 working groups, responsible for medical device biocompatibility standards on skin irritation, sensitization, and toxicological risk assessment. In addition, he serves on the National Toxicology Program's Science Advisory Committee on Alternative Toxicological Methods, SACADM, and is president of the SOT's In Vitro and Alternative Methods Specialty Section. Kelly holds a PhD in toxicology from the University of Minnesota, is a diplomate of the DABT, and a European registered toxicologist as well. Thanks for coming to present to us today, Kelly. Please take it away.
All right, thanks. Kirsty, can you hear me?
Yes.
Okay, I turned up my volume to make sure you could hear me. Anyway, thanks for the introduction, and thanks for our attendees for tuning in to listen to this quick update, I hope, on the world of in vitro irritation testing for medical devices. Here's my legal disclaimer. I always have to show these so Medtronic's lawyers won't get excited. Anything I'm talking about here is public domain information, nothing proprietary, and they're my opinions, and I don't really have any conflicts of interest. So 3 things I'm going to touch on in the next few minutes: our in vitro irritation project, some follow-up work that we did, and what's the status of the new ISO 10993-23 standard. So in vitro irritation project. It's always nice to have a timeline to put things into perspective, and this project got going in 2009. I remember I went with Mike, excuse me, actually to the ISO TC 194 meeting in Berlin, and TC 194 stands for Technical Committee 194. They're responsible, the working groups of that committee are responsible for the ISO 10993 standards, and they used to meet every other year, and now they meet every year. But this was the first meeting I went to, and it was in Berlin. And I can remember getting up at the Working Group 8 meeting. That's Working Group 8 is responsible for the ISO 10993-10 standard on irritation and sensitization. And I stood up and introduced myself. I work at Medtronic from Minneapolis, and I said, I think we should replace the rabbit test with a human skin test. And a lot of people on that committee said, well, why would you want to do that? The rabbit test works fine. But thankfully, a fellow named Wim de Jong, who was the convener of Working Group 8, he thought it was a good idea, and a few other people on the committee supported that concept. So that's how this whole thing began, and I was able to get a internal grant at Medtronic in 2010 to do a pilot project, and that took a couple of years. It was completed and published in 2013, and went back to Working Group 8, presented our results, and suddenly they thought it was a great idea, and and they encouraged us to do a validation study. So this round robin study began. Wim de Jong and I were the co-directors of it, and it took a lot of prep work, and I'll talk about that later, but eventually this study was completed and it was published. We did a follow-up study, and then this year the Part 23 standard is being finalized. We'll get to that. We'll get to that later. So just a second here. Okay, I wanted to show you, this is an old version of the 10993-1 biocompatibility matrix because I couldn't find a color version of the new matrix, but I just wanted to show you these, the way these tests line up. There's 3 tests required essentially for all medical devices. Doesn't matter, you know, if it's a tongue depressor or a heart valve. Those tests are cytotoxicity, sensitization, and irritation. Irritation is the one we're going to focus on today. You can see if you look down through this column, it's required for virtually every device that exists, regardless of contact time or type of contact. So when I first began looking at this issue back in 2009, I did some research and found that Medtronic's historical failure rate on this test was 1%, which was a good thing. This is the Dre's rabbit skin irritation test that is typically done. We had a long-term, since the 1980s, 1% failure rate, which is common throughout the device industry. It's rare to fail that test, which is a good thing. But on the other hand, I kept thinking, well, you know, we're running this test, consuming these animals, getting the same results. Essentially, there's got to be a better way of, you know, doing this. And so that was my pitch to Working Group 8 in 2009. Let's look for a better method. And at the time, I was aware that ECVAM, European Center for Validation of Alternative Methods, and published this famous study, a validation study they did that cost 1 million euros to complete. And they looked at 3 different tissues. They're listed here on the left, Episkin, Skinethic, RHE, and then Epiderm, made by MacTech. And the first 2 are made by L'Oreal. And this was published in Alternatives to Laboratory Animals in 2007. And they looked at about 60 different chemicals, and these were pure chemicals. And they decided, decided eventually, based on the results of this study and then some follow-up work, that these RHE tissues, which stands for reconstructed human epidermis, that they were acceptable replacements for the rabbit test. So that led me to produce this, or think about this pilot project, and our goal was just to determine whether or not these RHE tissues could be reliable alternatives for the rabbit skin test that we do. And the difference between this paper I mentioned, the 2007 paper, and what we're talking about is the ECVAM study tested pure chemicals, and medical device extracts aren't pure chemicals. They're dilute extracts, polar and non-polar solvent extracts of medical devices at this ratio that Mike mentioned. Typically, it's 3 square centimeters of surface area per mil. So the idea to go after a Quest grant, which is in the lower right-hand corner, started at a toxicology meeting in Baltimore in the spring of 2009, where I met Alan Goldberg, who is the founder of the Center for Alternatives Testing at Johns Hopkins. He introduced me to some people at CTOX, as he was on their board of directors, and we came up with a plan to do a little pilot project, and I applied for a grant, and I got it, a Quest grant at Medtronic. That project took a couple of years. It was pretty simple. We picked 2 skin irritants. We had 11 medical device polymers, and we spiked very low levels. And by spike, I mean we just titrated very low levels of lactic acid and heptanoic acid into polar and non-polar extracts of those 11 medical device polymers. And we tested them 100 microliter doses onto epiderm skin models. Epiderm is produced by MacTech in Massachusetts and in Slovakia. And our exposure time was 24 hours, and we used MTT, and we also used IL-1 alpha, looking for cytokines, but MTT is the key predictor. And we were able to show without a doubt that these epiderm tissues could identify low levels of these irritant chemicals in medical device extracts. And so that was published in 2013, and that was what helped convince Working Group 8 that we should do this round robin study. And eventually we had, to my surprise, we had 24 labs that volunteered to participate in our round robin study. And we were originally going to work with just epiderm tissues, but we eventually added skin ethic also. And L'Oreal joined the project, which was very helpful. And this took a lot of time. Key, I guess I'd say rate limiting step in this process was to find or make irritant polymers, because you can't just get on Amazon.com or go to Home Depot or someplace and buy irritant polymers. There's no real market for these things. Therefore, they're hard to find. So it was a lot of trial and error to make polymers. Here's my irritant slide, irritant polymer slide. They're hard to make. It was a lot of empirical work, and we were mixing them, casting them, and doing some hot melt experiments. And then we did extraction studies on them, tested the extracts by LCMS, and did some tissue testing also to confirm that whatever we were extracting from them, whatever levels of irritants we were extracting, were enough to produce a positive result in the human tissue tests. And these are just a couple pictures. On the left is a lab mixer we used. On the right are some Teflon blocks we used to cast our samples in. And those casts were 1 centimeter by 1.5 centimeters by a couple millimeters thick. So the total surface area was 3 square centimeters, which is our preferred surface area for extractions. So these are the materials we used. We had 3 non-irritants and 4 irritants that were made or purchased. The Y4 polymer came from Japan, and Y1 also, and the others we made on our own. So this was a 2-phase study, this validation study with epiderm tissues and the skin ethic tissues. We had 16 labs that participated in the phase 1, the epiderm phase. It was a blind study, coded samples, again, extraction, tissue dosing, and then MTT. And MTT is a yellow tetrazole dye you're probably familiar with, and it's the cells, if the skin cells are alive, it's a mitochondrial reductase assay, and so it turns the yellow dye purple, and then it's the spectrophotometer that measures it. And I call this 3x3 testing, which means that we did n equals 3 samples on 3 different days, so it would be actually probably 3 different weeks. So a total of 9 samples, blinded samples, were tested, and this whole project, there was 2,000 samples that were made and shipped, coded and shipped by Nelson Labs in Utah, which was terrific. Anyway, phase 2, we had 8 labs that participated. Same story: extractions, tissue dosing, MTT, and then the same 3x3 testing. I'm just going to show you one slide here of data, and I don't know if you can see my cursor, but on the left side, this is cell viability. And then this is the y-axis, x-axis is just these different samples. And if we look along here for a polymer, or for a device to produce an irritant response in this assay, you would have to have cell viability less than 50%. So the extracts would have to kill more than 50% of the cells, the human 3D skin cells. And we look, if we just go from left to right on here, you'll see these samples, 100% silicone, and then this polyurethane. Elastane, ADA, those are negative controls, and our cell viability is up here at 100%, which is what we expected. If we move to the right, you have PVC plus 4% genopol, and genopol is a surfactant. That's a really good, it's very cytotoxic, and it's a good skin irritant. And so we're seeing, and this is one of our positive samples, so here we're seeing cell viability below 50%, so definitely it's an irritant. Next sample is silicone plus SDS, sodium dodecyl sulfate. That's a positive control. It's used for human patch testing and in the rabbit test. And without a doubt, we're seeing, only 10% cell viability here, so 90% kill rate. So that's positive. Next one over, we're seeing it's silicone and 25% heptanoic acid. Heptanoic acid is a strong irritant, and it's a corrosive. So you might be wondering, why are we seeing 100% cell viability here? And the reason is heptanoic acid is not very soluble in saline, and this is the saline extract that we're seeing. So if I showed you the slide for sesame oil, which is our nonpolar solvent, you would see the cell viability would be down here in the positive range for this particular one. Next sample is Y1, another negative control, and cell viability again is 100%. And last one is Y4. That's PVC with genopol in a slightly different type of genopol than was in the previous PVC sample. And again, down here, we're seeing very low cell viability, so very positive result. So here's a quick summary of the results from epiderm. And the thing to look at is over on the right side, the overall accuracy in predicting, what these things were. And we're seeing well over 90% accuracy for this particular tissue. And if we go to the next one, excuse me, I'm having trouble shifting my slides here. Okay, here's the next one. This is skin ethic RHE. Same thing, just look to the right and you'll see we're way up around high 90s to 100% overall accuracy in predicting what those samples were. So this work was summarized in 6 papers on an editorial, so I guess 7 papers that were published in a special issue of Toxicology In Vitro in 2018. And these are all open source papers. You can read them if you wish. They're available online. Our conclusions, though, were from this study that these RHE tissues are definitely robust models for detecting low levels of irritants in medical device extracts, and therefore we believe that they were acceptable replacements for the rabbit test. So let's go to our follow-up study. This is some work that was done in 2019 and in 2020. We had 2 laboratories that participated, and we just wanted to try some other tissues. And the first lab is the Shandong Quality Research Center. It's part of the National Medical Products Administration in Jinan, China. And you see a picture of Mike Wolf and I here in front of the Jinan Center. And this is Chenghu Liu to my left, who's the director of biocompatibility there. And we went to visit him in January last year. And they were going to test skin ethic tissues that are made in China. They're grown in Shanghai because they were going to participate in the original round robin studies, but the tissues never got to them. They got caught up in Chinese customs, and so they weren't viable. So they wanted to participate in this follow-up study, and we were just happy that they would help us because they did some great work. The other lab is called Cell Systems. It's near Cologne, Germany, and they have a tissue, an RHE tissue called EpiCS, Epi Cell Systems, and this is the city Tronsdorf, that's near Cologne. And so we made up identical sets of polymers, we used, similar to the polymers that were used in the round robin study, and sent them to these labs. Nelson Labs did all this blinded samples again. And the Jinan lab, at the same time they were doing the RHE tissue testing, they took another set of polymer samples, extracted them, and did intracutaneous testing on rabbits with the same samples. So excuse me, we gotta go next slide. So this just came out a few weeks ago in Toxicology In Vitro. Again, this is an open source paper, and this summarizes the work from the follow-up study. And I guess I could, in a nutshell, say that these 2 labs, the follow-up lab in China and then in Germany, testing the same samples, essentially got the same results that we did in the big round robin study. And also the Chinese lab in Jinan, their results for the intracutaneous testing with rabbits were the same as the results they got from the RHE tissues, which was what we expected, and that was helpful. So there's some other information in this paper that summarizes some other work, intracutaneous RHE testing that was done during the actually before the round robin study. And there's another set of data that we got. We call it the dual data study. There was 45 medical devices or medical device materials that were tested on RHE tissues. The RHE tissue test was done on them. And then they were also subjected to the rabbit intracutaneous test. And the RHE tissues produced the same results for all those samples as the rabbit test did, which was more confirmation that the RHE test is comparable to the intracutaneous test. So last part of the story, the 10993 standard, this has gone through a couple of different rewrites over the years. It went through all the standard steps that ISO does. You have a working draft and then a committee draft, and then you have what's called a draft international standard and a final draft international standard. And along the way, there's the people that review it, the members of ISO TC 194 get to submit comments and then vote on it. And every one of these received a unanimous vote until we got to the FDIS standard. And the reason we had one no vote is somebody was on vacation and who was filling in for him didn't know what to do, so they voted no, unfortunately. But this new standard, it still it has all the information about this in vitro study, and then it also still contains the rabbit information, the rabbit testing information, because we figure there's going to be a transition period between the time this standard is published and the availability of these RHE tissues around the world. So the standard, it says that it recommends, or it says they shall, people shall use the in vitro assay before considering, doing animal testing, or if you're going to do human batch testing, which is rare. And we think eventually this will be accepted worldwide. We're also pursuing FDA with MDDT submission, and hopefully finish that up in the next few months. What else can I tell you? We talked to some people at TUV recently about this, and they're excited about it. And their thought was that 2021 will be a transition year, because this -23 standard, this should be published either this month or in January. And I guess that's all I have on that. And what is next here? Oh, I wanted to just take a couple minutes here and show you, as I mentioned earlier, Wim de Jong and I were the co-directors of this round robin study, but we were not the only ones involved with this. There was dozens of people that helped with this. We had 23 different organizations that participated, and the ones that are highlighted here, I just wanted to give them a shout out. Arthrex in the US and Germany played a critical role in developing polymer samples, and they also provided the dual data information that we had. Episcan Academy, this is L'Oreal in France, they were involved with a great deal of pre-validation work, testing samples, and also providing free training to labs that participated in the round robin study. MacTech in the US, and then MacTech In Vitro Life Sciences in Slovakia, and provided training for the laboratories. And MacTech in Slovakia did an immense amount of pre-validation work. We couldn't have done it without them. NAMSA in the US, they did provided a lot of support for us behind the scenes, doing rabbit testing and tissue testing early on. RIVM, Wim's lab, did an immense amount of pre-validation work, testing polymer samples on tissues. And Nelson Labs, they did sample prep and shipping for the 2,000 samples for the round robin study and the follow-up study. So again, critical people that participated in this study. And then SEH Consulting is Sebastian Hoffman in Germany. He was our biostatistician, and we couldn't have done this without his help either. And here's a name, here are 31 names, and I hope I've got most people here. And again, I just wanted to give a brief shout out to Wim de Jong again, co-director of this project. It never would have happened without him. Great guy, did an immense amount of work on this. Elena Kandarova was the director of MacTech's lab in Slovakia, did a huge amount of the pre-validation work, testing polymers, provided all kinds of free tissues for us. Sebastian Hoffman, our statistician, he helped with the protocols and then analyzing all the data. Michelle Lee and Audrey Turley from Nelson Labs, handling the sample prep and shipping. They were terrific. Christian Pelavision from Episcan Academy, again, an immense amount of work that he and his colleagues did to make this thing happen. Yuji Hashima from the National Institute of Health Sciences in Japan. They did a great deal of work, and they were one of the few labs that tested both polymers, or excuse me, both types of tissues, and also provided the Y4 or Y1 polymers for us. Bo Rollins and Nicholas Cristiano used to work together at Arthrex, and they were the dynamic duo that developed several of our irritant polymers. I know we're running out of time here, so I don't have time to mention all these other folks. Most worked for contract labs, but they're all important. And this never would have happened without their help and support. And this project took, thousands of hours of people's effort over many years. And the time and money for this was all donated by these people and their companies. So can't thank them enough. Lastly, what else do we have? Oh, references. And if we could make a PDF of these slides available to you if you're interested. But as I mentioned earlier, our first study, our pilot project came out in 2013. This is in In Vitro, Toxicology In Vitro, and that I don't believe is open source, but the whole round robin study is available. It's open access. You can find it easily on the In Vitro website. And then our follow-up study that just came out is also available on Toxicology In Vitro's website. And that's it for me. Thanks for your attention.
Wonderful. Thank you, Kelly. I have to say there's also compliments coming into you, the same really appreciation to you and all the team, all the people that were involved in this work. Couple of questions already in the Q&A, and so anyone has a question, feel free to type it in. The first one is that, so the 10.99323 draft describes the in vitro test with a focus on polymer extracts, but how would neat liquid medical devices be tested using this method?
I think they'd have to be diluted, actually, if you're gonna do that. And I guess I'm gonna think of what would be a neat liquid medical device, some solution. But if you look back, the key study that was done by ECFAM that I mentioned, that 2007 study, it's in Alternatives to Laboratory Animals, ETLA. They tested about 50 chemicals in that study. There were, they're half irritants, half non-irritants. There are 38 irritants, and those were tested neat. They were pure. They weren't diluted, and they used 25 microliters to dose their tissues, and they only had a 15-minute exposure period. Because as you might imagine, if you had some really strong irritant that was a pure irritant, you wouldn't need to have a high level dose. Like in our validation study, our dosing level was 100 microliters, so it was 4 times as high because we were dealing with these dilute medical device extracts, and we used exposure times of 18 to 24 hours compared to the 15-minute exposure time that ECFAM used for those pure chemicals. And I guess conceptually, if you think about it, if you have a pure irritant, you're using a small amount of it and having a short exposure time, and that's enough to show you it's an irritant, versus these medical device extracts are dilute chicken soup solutions, I guess is what I would call them, and it takes a longer period of time, a higher volume in a longer period of time to show whether or not they're irritants. And the reason, I guess I should say the backstory on why we did this longer exposure time was because there was quite a history with these RHE tissues in the cosmetics industry, and they just learned from experience that they had to use higher volumes, dosing volumes on the tissues and longer exposure times to detect low levels of either strong irritants or just mild irritants in cosmetics. So if you're talking about neat medical devices, I would say you'd probably want to use them in the same tissues, but maybe go back to that old ECFAM protocol, which would be a lower dosing level, maybe 50 microliters or 25 microliters, and a shorter exposure time. So long-winded answer.
And actually, Helena has piped in to confirm what you just said about cosmetic materials, which are tested in the same type of protocol. Yeah.
She's the oracle, so great. Perfect, thanks.
There's a question about acceptance by regulatory agencies. So once the ISO standard is published, do you have any sense of FDA, EMEA, or the Chinese regulatory agencies, whether they'll begin accepting this method?
Well, notified bodies are excited about it in Europe. Everyone that I've talked to says, yes, they're ready. They keep saying, when's that thing going to be published? China, in our picture, there was a picture of Chenghu Liu, who Mike and I know, because he came to the University of Minnesota and worked in Mike's old lab, the blood lab, for a year at the University of Minnesota. Great guy, but we've talked to him about this in the past, and he said once that's published, that will become the rule of law in China, which is great.
Mm-hmm.
Japan, I'm optimistic that the PMDA will accept it. I know the National Institute of Health Sciences in Japan participated in this study, and they believe in it strongly. FDA is still somewhat skeptical, and that's why we're working with them through this MDDT process. And I think one of the reasons they were skeptical, or two reasons, was whether or not these RHE tissues truly produce results that are comparable to the intracutaneous test in rabbits. And I think we've shown that now in our follow-up study. And there's also there's another paper from Japan, this Nomura paper from 2018, that had show the same thing. They were using epidermal tissues and testing various irritant polymers, and then doing intracutaneous testing with them also, and getting the same results. So I think that answer, hopefully those two things will resolve that issue. And the other question they had is they were just wondering about, FDA was wondering about mild irritants, and they didn't think that these tissues could detect mild irritants, that was actually true in this 2007 ETLA ECFAM paper that I mentioned earlier. Part of one of the early goals of ECFAM's work, the World Health Organization asked them if these tissues could identify, mild irritants, so they could have a label. They could label. Use labels on chemicals that would say mild irritant. And in the original 2007 study with 25 microliter dosing, and then the showed exposure time, they said that those tissues could not identify mild irritants. However, as I mentioned, and Helena's mentioned in her notes, we've been able to show, well, the cosmetics industry has been doing this for years, and now with medical devices, we've been able to show that without a doubt, if you use 100 microliter dosing levels and then long exposure times, you can identify anything that's, if it's, as Helena says, if it's any kind of irritant, it will be identified if you use this high dose level and a long enough exposure time. So those are two issues that FDA was concerned about. We hope that we'll be able to show that to them, and we'll see where it goes with them. But we're optimistic. But otherwise, anybody else we've talked to has been really positive about this.
That's good. A brief question, maybe, do you need to perform in vitro irritation studies under GLP? And I wonder if that depends on the context. But
if it's for a regulatory submission, I would. And most of these, I think virtually all the contract labs, that exist in the world, that work for the medical device industry, they're all GLP labs, and they probably have other certifications like ISO 17025, and there's probably others. But for submissions, I think that's, well, at least in the U.S., I think that's definitely important.
And maybe one last question about which I think may or may not go beyond medical devices, but we'll see what you think. Is there a plan to develop in vitro models for moderate or severe irritant categories?
I don't think so. I don't think that's necessary. I mean, these assays already exist for corrosives.
Yep.
And there's a OECD 439 is the guidance document for tissues, RHE tissues for these human tissues. It's OECD 439. A new version of it just came out, and that's for irritation testing. There's another one, and I'm forgetting the number of it, but it's OECD 4 something, and that's for corrosives.
It might be 431 if I'm remembering correctly.
Yeah, well, thankfully, the device industry, we don't see corrosives, as far as I know. And that's just something, I mean, it's rare to fail the irritation test. And corrosives are something that the ISO standards have never required that, because, if you think about it again, conceptually, if you're doing extracts, you would have to have something incredibly nasty, I think, in a medical device that would come out at a high enough level to produce a corrosive response.
That's why I was thinking maybe it was going beyond medical devices, because there are, and just to confirm for the questioner, for the chemicals and pesticides, there are OECD test guidelines to cover those, as you said.
Maybe kind of tangentially to that, one other thing I didn't mention is that this -23 standard, the new one that's coming out, is for, I'd say most traditional medical devices, but it's not for like ocular irritation, for like contact lenses, for example, or for gastrointestinal things. And eventually, I mean, there are annexes in the -23 that mention those, and there are some, human tissue assays that have been developed for these other types of, mucosal, for instance, tissues. And someday I hope that those are validated for, contact lenses or for other kinds of, oral devices, or like penile or vaginal or other things that these particular RHE tissues haven't been validated for, even though they might work for that, for those endpoints. They haven't been validated for that, and so that's for some future group of people that are going to be members of the working group 8, probably after I retire.
Well, I can just mention that Helena mentioned that they are testing contact lenses directly on the tissues. So that's
Good.
Something that maybe will be
Okay.
coming.
We got to get somebody to validate those.
Yeah.
Working. Actually, there's some people in Korea that are doing a study on that right now. I forgot. At one of the universities in Korea.
Okay, well, I think we'll go ahead and finish up the webinar. Thank you all for your questions and discussions, and thank you to Mike and Kelly for the really nice presentations. Really appreciate your time preparing and being with us today, and we'll look forward to some more webinars in the new year, and I hope everyone has a happy and healthy holiday.
Great. Thanks, Christy.